In this video, you will be watching an overview of laboratory safety and precautions necessary for working in the microbiology laboratory. You will also watch a demonstration of aseptic technique and the process of serial dilution, which is the first step in the enumeration of microorganisms, and finally how to conduct a total plate count. Safety is an important consideration in any laboratory environment. Whilst the microorganisms you will encounter in the practical sessions are not pathogenic, it is important to observe the following procedures and safety points at every laboratory session. On entering the laboratory, wash your hands. Laboratory coats are provided for your use during the practicals and must be worn, correctly fastened. Long hair must be tied back during practicals as this is a potential hazard when working over a Bunsen burner. Listen carefully to the instructions given to you at the beginning of the practical. If you are in any doubt, please ask the lecturer or technician. Be aware of the locations of fire extinguisher, eye wash station, first aid kit and emergency exits. Inform the lecturer or technician of any accident, spill, or potential hazard. Never put anything in your mouth in a laboratory. This is a major route for microbes to enter the body. No eating or drinking is allowed in the laboratory. This includes chewing gum. At the end of your work, clean the worktop using a disposable paper towel and the D10 detergent disinfectant provided. First, spray sufficient amount of the detergent disinfectant onto the worktop, wait for 5 minutes and wipe with the paper towel. Before you leave the laboratory, remove your lab coat first, then wash hands thoroughly. Always wash your hands when you leave the laboratory. Coats must be left in the ante-room as you leave and must not be worn in any other location. We will continue by looking at aseptic techniques. Aseptic techniques. The word aseptic derived from sepsis means infection or infectious material, a means without. This is an extremely important aspect of microbiology laboratory procedure. Aseptic technique allows one to work with microbes without contaminating the worker or workstation, and without getting extraneous environmental microbes mixed up with those being studied. Aseptic technique must be observed in every practical, and includes the use of a Bunsen burner blue working flame. Metal loops and bottlenecks must be flamed before and after use. Never place a sterile item on the bench surface or touch with the hands. Only remove sterile disposables from pack when needed. Never touch pipette tips or invert pipettes when in use. Do not leave lids off petri dishes or place on bench. This will avoid contamination from airborne microbes and spores. Now that you have seen how to practice aseptic techniques, we will continue to look at the process of serial dilution. Serial dilution is a process of stepwise dilution of a solution, with an associated dilution factor, performed to convert a dense solution into a more usable concentration. Bacterial cell numbers are very high in food samples. Plating out the original food sample would just lead to the growth of bacterial lawn, a smear of many individual colonies. The bacterial cells is reduced by repeatedly diluting the original sample, using a diluent, e.g. Ringer's solution. A small amount of each diluted bacterial sample is then spread onto an agar plate. The number of bacterial colonies that grow are then counted. The number of bacteria in the original sample is determined by working backwards using multiplication with the dilution factor. In the next demonstration, we will perform a serial dilution from an original liquid sample of a bacterial culture. We will then plate the diluted sample onto an agar surface to enumerate the bacterial numbers in the original sample. To perform a serial dilution, we need the following. Original sample to be diluted. 9 ml diluents, in this case Ringer's solution, in dilution bottles. Vortex mixer. 1000 microliter micropipette with corresponding pipette tips. In the serial dilution we will be using dilution factor of 10. This means, we will transfer 1 ml of sample into 9 ml of diluent to give a total of 10 ml. In other words, the diluted solution will contain 1 part of the original sample in 10 part of the whole solution or 9 part of the diluent. The original sample, which is undiluted, is represented as 10 to the power 0. Now label the bottles with the respective dilution levels. That is, 10 to the power -1 for the first dilution bottle. 10 to the power -2 for the second dilution bottle, 10 to the power -3 for the third dilution bottle, 10 to the power -4 for the fourth dilution bottle, and so on depending on the level of dilution required. To begin the dilution process, mix the sample thoroughly using the vortex mixer. Next, open the cap and flame the neck of the bottle. Using the 1000 microliter micropipette, aseptically transfer 1 ml of the culture from the sample bottle into the first dilution bottle. This gives us a dilution level of 10 to the power -1. Mix very well using the vortex mixer. Next, open the cap and flame the neck of the bottle. Aseptically pipette 1 ml or 1000 microliter of culture from the first dilution bottle into the second dilution bottle using a fresh pipette tip. This gives us a dilution level of 10 to the power -2. Replace the cap of the second dilution bottle and mix very well using the vortex mixer. Open the cap and flame the neck of the bottle. Using a fresh pipette tip, transfer 1 ml or 1000 microliter from second dilution bottle to the third dilution bottle. This gives us a dilution level of 10 to the power minus 3. Replace the cap, mixing and flaming as before. Repeat for rest of the series. Remember to always flame the neck of the bottle after mixing and opening the cap. Always use a fresh pipette tip to transfer the sample. The respective dilutions are now ready to be plated. The next step is plating the diluted samples onto agar surfaces to be incubated and subsequent counting. Before we start, the agar plates must be labeled with the dilution levels and any ID for identification. There are two ways of determining the dilution levels on the plate, which depends on the amount of sample transferred from the diluted samples. In the first instance, if a 1000 microliter or 1 milliliter is transferred, then the dilution levels labeled on the plates remain the same as those on the dilution bottles. In the second instance, if a 100 microliter or 0.1 milliliter is transferred, then we are further diluting the sample by a factor of 10. The dilution levels on the plates will therefore become one dilution level higher than that on the dilution bottle from which the sample was taken. That means, a dilution level of 10 to the power minus 1 on the dilution bottle, will become 10 to the power minus 2 on the agar plate, if we transferred 100 microliter or 0.1 milliliter of the sample. There are two options of plating the diluted samples. The first option is to plate starting from the least diluted sample. In this case the least diluted sample is 10-1. This is the sample in the first diluted bottle, which is also our first dilution. For this option, pipette tips must be changed to a fresh one after each sample transfer. Using a 100 microliter micropipette, aseptically transfer 100 microliter or 0.1 milliliter of the first dilution onto the agar plate labeled with the 10 to the power minus 2. Second dilution to 10 to the power minus 3 agar plate. Third dilution to 10 to the power minus 4 agar plate, and so on, making sure a new fresh pipette tip is used for each transfer. In the second option, plating starts from the more diluted sample. In this case 10 to the power minus 7. For this option, the same pipette tip can be used to transfer the sample from respective dilutions to corresponding agar plates. The next step after transferring the sample to the agar surface is to spread the sample to cover the agar surface. This is done using a sterile spreader. Like the two plating methods, there are two methods of spreading. The first option is starting from the less diluted sample, that is 10 to the power minus 2. In this case, a new spreader must be used for spreading the sample on each agar surface. The The second option is starting from the more diluted sample. In this case, the same spreader can be used for spreading the sample on the agar surface, starting from the more diluted sample. After plating, the plates are incubated at respective temperatures and time before observation and enumeration. Counting. To count the colonies formed on the plate to determine the bacteria number, plates to be counted in the series should have between 30 and 300 colonies. Fewer than 30 colonies are not acceptable for statistical reasons, too few may not be representative of the sample. More than 300 colonies on a plate are likely to produce colonies too close to each other or covering other colonies. A colony counter is used for counting the colonies on the plate. You need a marker pen to mark colonies as you count. The colony counter is designed to beep to touch and record the number as you count. Switch the counter on and place the agar plate on the counter with the agar side up and press reset. Touch colonies lightly using the marker pen, making sure you hear a beep at each touch. The number of colonies is recorded at the end.
Okay.
The colony count at dilution level 10 to the power minus 5 is 311, which is too numerous as explained before. The colony count at dilution level 10 to the power minus 7 is 5, which is too few. Therefore, the colony count to use to calculate the bacteria number is the count at dilution level 10 to the power minus 6, which is 43. The following formula can be used to calculate the final bacteria count in the original sample. That is, the number of colonies counted, divided by the dilution level plated, multiplied by the amount of sample that was plated. Count is represented as colony forming units, CFU per unit of tested sample. The bacteria number can also be calculated and expressed in log format as log CFU per unit of the sample taken. The above formula can be used for such calculation. You have now reached the end of this demonstration. You may watch it again when needed.